snowflake landed on a glass surface and began to disappear. To an ordinary camera, the event would look simple. Its sharp branches would soften, a thin layer of water would spread outward, and within moments the crystal would be gone. But a new imaging system saw something no conventional camera could capture. It recorded changes in the snowflake's thickness measured in nanometers, traced the three-dimensional shape of every arm, and followed a film of meltwater so thin that it was almost invisible. The most surprising part was not the snowflake. The instrument had no traditional camera lens. Instead, researchers built a compact imaging chip that scrambles incoming light, then uses mathematics and artificial intelligence to reconstruct a detailed three-dimensional video. The system can capture an area several centimeters wide at 30 frames per second while producing images containing roughly 1 billion pixels. That combination attacks one of microscopy's oldest problems. Scientists usually have to choose between seeing a large area and seeing extremely small details. A microscope can reveal the structure of a single cell, but it may miss what is happening across a larger population. A wider view captures the group, but individual details disappear. The new system is designed to do both. To understand how, imagine photographing an entire football field while still being able to examine the texture of a single blade of grass. Ordinary cameras struggle because magnification reduces the visible area. Higher detail usually means a narrower field of view. The researchers approach the problem from a different direction. Instead of using glass lenses to focus an image directly onto a sensor, they used a technique called tachography. In tachography, light passes through or reflects from a sample before producing a complicated interference pattern on a detector. The raw pattern does not resemble a normal photograph. It looks more like overlapping waves or distorted shadows. But the distortion contains information. If a computer knows how the light interacted with the imaging system, it can work backward from the recorded pattern and reconstruct the object that created it. The method can recover not only brightness, but also the phase of the light. Phase reveals how far different parts of a wave have traveled. When light passes through a transparent object such as a cell or a crystal of ice, thicker areas delay it more than thinner areas. By measuring those delays, the system can calculate the object's height and build a three-dimensional map without cutting it open or adding fluorescent dyes. Tachography already existed, but it came with a major limitation. Producing a large, detailed image often required many separate measurements. That worked for objects that remain still. Living cells, melting ice, and growing bacteria do not wait for a scanner. By the time a conventional system completed its measurements, the sample might have moved, changed shape, or disappeared. The new method turns that movement from a problem into part of the solution. Researchers develop what they call a spacetime neural field. Rather than reconstructing each video frame independently, the AI model treats the entire recording as one continuous object changing through space and time. Adjacent frames share enormous amounts of information. A cell does not become a completely unrelated object 1/30th of a second later. Most of its structure remains similar, while only certain regions move or change. The model learns those relationships. Information from one moment helps improve the reconstruction of another. Each measurement therefore contributes to both spatial detail and motion over time. The result is video rate gigapixel tachography. To demonstrate the system, the team filmed a melting snowflake. The reconstructed images displayed colored bands across the crystal. These were not the natural colors of the ice. Each band represented a different thickness, creating something like a topographic map of a frozen landscape. As the snowflake melted, the bands shifted across its branches. A nanometer-scale film of water spread over the surrounding surface before the main structure visibly collapsed. The demonstration was visually striking, but snowflakes were only the beginning. The researchers placed the compact sensor inside a cell culture incubator and recorded living bone marrow stem cells. Traditional high-resolution microscopes often require researchers to remove samples from their controlled environment or illuminate them with intense light. Fluorescent imaging may also require dyes or genetically modified molecules that glow. These methods are extremely useful, but they can alter the sample, damage cells over time, or reveal only the structures that have been labeled. The lensless chip watched the cells continuously without fluorescent markers. It mapped their three-dimensional shapes and followed them as they moved across the surface. The team then created a scratch through a layer of cells, a common experiment used to study wound healing. Cells near the empty space began migrating inward. The system recorded the population as it gradually closed the gap. But the wider field of view did not erase the smallest details. In magnified regions, the reconstructed video revealed delicate tunneling nanotubes connecting neighboring cells. These structures are extremely thin extensions of the cell membrane. Cells can use them to exchange molecules, organelles, and signals. They are difficult to monitor across large populations because a microscope must combine high resolution with continuous observation. This is where the new system scale becomes important. It can watch broad behavior, such as thousands of cells moving together, while preserving enough detail to investigate individual connections. The researchers also observed colonies of E. coli bacteria. Under normal conditions, the bacteria divided and expanded across the imaging area. After exposure to an antibiotic, some cells stopped dividing normally and stretched into long filaments. That change is more than an interesting shape. Bacterial growth patterns can reveal whether a drug is affecting the organism. Faster, label-free imaging could eventually help researchers study antibiotic responses and investigate resistance without waiting for conventional laboratory tests. In another experiment, the system examined dissolving microneedle patches. These patches contain arrays of microscopic needles designed to enter the outer layers of skin and deliver drugs with less pain than an ordinary injection. Their performance depends on shape, consistency, and the speed at which the needles dissolve. The imaging chip created three-dimensional maps of the arrays and followed their structures as they changed. One instrument had therefore recorded melting ice, healing cells, bacterial growth, and dissolving medical devices. The same principle could reach even further because lensless imaging is not limited to visible light. Traditional lenses become difficult or expensive to build for some wavelengths. The researchers tested their reconstruction method using extreme ultraviolet light at a wavelength of 13.5 nanometers. This is the same region of the electromagnetic spectrum used in advanced photolithography systems that manufacture leading computer chips. Extreme ultraviolet beams can fluctuate in shape and intensity. Those changes normally complicate imaging. The new approach reconstructed both the object and the changing illumination using fewer measurements than conventional methods. That could matter wherever radiation exposure must be limited. In biological samples, excessive radiation can cause damage. In industrial systems, faster reconstruction can reduce inspection time. However, the technology is not a magical replacement for every microscope. The billion-pixel video is computationally reconstructed rather than captured as an ordinary photograph. Its accuracy depends on the physical model, sensor calibration, and quality of the measurements. Complex samples may scatter light in ways that are harder to interpret. Processing such large datasets also requires significant computing power. A practical clinical device would need to reconstruct results quickly, operate reliably, and prove that the details it reveals correspond to real biological structures. The demonstrations show capability, not immediate medical diagnosis. Yet the larger shift is already clear. For centuries, microscopes improved mainly through better lenses. Scientists polished glass more precisely, corrected optical distortions, and combined multiple lenses to increase magnification. Computational imaging changes that relationship. The physical hardware can become simpler, while more of the imaging work moves into software. The sensor does not need to create a recognizable picture by itself. It only needs to record enough information for an algorithm to reconstruct one. This could make high-resolution imaging devices smaller and easier to place in environments where conventional microscopes cannot operate. A compact chip might remain inside an incubator for days, continuously recording cells without repeatedly moving the sample. Researchers could watch cancer cells respond to treatment, observe tissue regeneration, follow infections, or compare thousands of drug candidates. Instead of receiving a few isolated snapshots, they could study the complete sequence of events. That difference is similar to replacing several photographs of a storm with a satellite video showing how the entire weather system develops. The individual images are useful, but the motion reveals relationships that static observations miss. The melting snowflake captured the same idea on a smaller scale. It was not simply photographed before and after melting. Its changing thickness became a moving three-dimensional landscape. A crystal only millimeters wide revealed shifts measured at less than 1,000th of a millimeter, across a field large enough to observe the complete structure. The snowflake vanished within moments. The imaging system preserved the process. And that may be the real importance of the technology. Science often studies objects by freezing, staining, cutting, or isolating them. Those methods reveal extraordinary detail, but they can remove the very movement researchers want to understand. This lensless chip attempts something different. It watches large, living and changing systems while still resolving their smallest structures. The first demonstration followed ice turning into water. The next generation may follow cells becoming tissue, bacteria responding to drugs or diseases changing inside the body. A camera without a lens has learned to see both the forest and the trees and to record what happens while they are still moving.